Hello everyone, welcome to the first video in E for Education. In today's video, we are going to discuss about the effect of nanoparticles like fullerene on fullerenol on proteins like ribonuclease A. So this article came out in acsomega in 2018 from Professor Swagata Dasgupta's lab in IIT Kharagpur. So this, as seen from this abstract from the figure, the fullerene and fullerenol acts on this protein, the RNase A or the ribonuclease A, and it shows some changes. So what are the changes that has been measured, or what are the effect of these molecules and protein has been measured are as follows. Number one is the inhibitory effect, and number two is the antioxidant effect. Okay, so let's dive into the paper and see what it deals with. The initial introduction part deals with the basic introduction of nanoparticles and their effect and what are the advantages of nanoparticle. In the second part, it deals with the fullerene and fullerenol. Why fullerene, which is one of the allotropes of carbon, generally known as C60, is quite important. Okay, and it actually shows in this paper that fullerene is required as sort of an antioxidant property. Due to its antioxidant property, fullerene can actually capture sort of free radicals, and that's why its role in biological system is slowly getting importance. Now, apart from fullerene, there is another substance, or rather its hydroxylated moiety called the fullerenol. Now, this fullerenol actually has multiple hydroxyl group. As you can think about that, fullerene being a completely hydrophobic moiety might not able to interact with RNase A. That's why researchers have developed this fullerenol, which has like multiple hydroxyl groups. And in this paper, they have tried to compare the effect of this molecule without any hydroxylated group as well as the hydroxylated group on this RNase A protein. Now, talking about this protein, so what this paper shows that this is a protein which is generally associated with cleaving of RNA and is one of the very common protein within our body. And this protein has 6 tyrosine residues. So these are different tyrosine residues. It has 6 tyrosine residues and no tryptophan. And this tyrosine residue serves as one of the important outcomes in this paper. So as we are discussing, this is fullerene, generally known as C60, and this is fullerenol with like different hydroxyl group attached to it. Now coming to the result and discussion part directly. In the initial part, they have tried to characterize the fullerenol. So in this paper has done some interesting work that they have converted fullerene to fullerenol in the lab using some reaction, and they have tried to compare or rather characterize this fullerenol formation. So what are the things they have tried to understand? They have tried to understand whether the attachment of hydroxylating group like OH groups have occurred or not, number one, and number two is how many hydroxyl groups are present. So that is quite interesting. Now coming to the resultant discussion, so the first thing is that they wanted to characterize the aqueous suspension of fullerenol. Initially, they have used UV studies, and these UV studies are generally shown in the SI. So we can just refer to the SI to see like what are the general sort of absorbance spectra for fullerene and fullerenol. Now the important thing is the FTIR studies from this paper, where they have shown the appearance of this peak around 1107, which is for fullerenol, suggesting that OH groups are attached to it. However, fullerene, there is nothing around like 1107, suggesting that this is actually for hydroxyl groups. So the fullerenol has been prepared. The second thing, they actually use the MALDIT, or rather the mass spectroscopy, to understand the number of OH groups. And from here, they have come to the conclusion that there are at least 30 to 40 OH groups attached to it, since they have calculated the mass from here, and from the mass they have tried to understand the number of OH groups. After characterizing after confirming that fullerenol has been prepared, they have shifted to studying the morphology, size, and the zeta potential at the charge on the this nanoparticles. So as expected, the fullerenol, which is shown in yellow, has a size radius around almost like around 200, more than 200 like that, whereas for fullerene it is like around 150 nanometer. So fullerene has a size much lower than fullerenol, as we can see from this picture, that due to attachment of the hydroxyl group, this size actually increases. Now once they have characterized it, so there are other like characterization like the UV which are discussed in the supplementary information, which I am currently not covering. Now they have tried to understand the interaction studies of fullerene and fullerenol with ribonuclease A. And in terms of interaction study, they have used fluorescence quenching studies. Okay, so the fullerene, the important thing is that they have not used any sort of absorbance or fluorescence spectra for fullerene. Rather, they have used the like emission spectra for tyrosine radicals, since ribonuclease A as tyrosine, sorry, not tyrosine radical, the tyrosine residue, which is present in ribonuclease A. It doesn't have any tryptophan, so that's why they have used tyrosine, which shows a peak around like 310 nanometer, the emission one. Now with the addition of fullerene and fullerenol, they have seen that the fluorescence spectra decreases, and from here they have tried to understand the binding value. They have used normal sort of Stern-Volmer equation to understand the KSV, and okay, the observation out from here is that the fullerene shows the Kb around 5.3 and fullerenol 3.5, which is a bit odd, since fullerenol, which has hydroxyl group, should have interacted with fuller protein to the better extent, but they have shown that fullerene, which is completely hydrophobic, like if interacts almost like 1.5 to 2 times more than that of fullerenol. So the reason they gave is like the bit like fullerene can interact with the protein using the like pi-pi interaction, since it is hydrophobic and protein has hydrophobic residues in its active site, whereas in fullerenol the pi-pi or the hydrophobic interaction is somewhat disturbed due to the like polar hydroxyl group. The next thing to try to understand is the inhibition studies using the agarose gel based assay, and they have used some like agarose gel assay where they've tried to understand that inhibition properties, and they have seen that the fullerene can actually inhibit the protein as well as fullerenol, and they show different types of inhibition. One is the non-competitive and is the mixed. Now the extent of inhibition, the Ki value for fullerenol is almost like 6 times higher, or rather the Ki is 6 times lower than fullerene, suggesting that fullerenol can inhibit the protein to a higher extent than fullerene. Okay, now the next thing is that the docking study. For the docking study, they have tried to understand the site of attachment of the fullerene and fullerenol to the protein. So this is some beautiful picture where they have seen that the interaction of fullerenol and fullerene, so the negative value, so this is delta G negative minus negative, the negative value for fullerene is much higher than that of fullerenol, which is also found in the experimental studies that fullerene binds to a greater extent than fullerenol. So the greater delta negative value from theoretical studies also supports the experimental result. And the structure 1 and 2 are the two different positions of attachment of fullerene and fullerenol. Now here it can be seen that fullerene and fullerenol attaches to a common position apart from the active site. So this is actually the active site, okay, where fullerenol attaches, and fullerene attaches away from the active site. Since fullerenol attaches towards the active site, so it can like prevent the substrate from binding to the active site, thereby it sort of serves a better inhibitor as compared to fullerene. But fullerene and fullerenol binds here, so they have sort of speculated that this region might be the allosteric site, or rather a different site where fullerene and fullerenol can bind to the protein. And these are the nearby residues around the fullerene and fullerenol which are present in the protein. Now, one of the important things they have come across in this paper is the prevention of dityrosine bond. So the dityrosine formation is actually the formation of sort of dityrosine, or rather two tyrosine joint comes close to one another and they form a covalent linkage. So this dityrosine has an indicator of oxidative stress. So dityrosine is present in sort of peptides, like during Alzheimer, dityrosine is found largely, which suggests that it is a sort of indicator or a biomarker of oxidative stress or related diseases. And this paper actually shows how they have sort of initiated a dityrosine formation. And now what happens that due to dityrosine formation, protein dimer is formed. Now we'll come to that a bit later. Talking about a dityrosine formation, a dityrosine shows some fluorescence, and the fluorescence emission is observed around like 405 to 410 nanometer for the DT or the dityrosine. Now what we can say is that, or rather the paper shows that in presence of fullerene with increase in concentration, the dityrosine emission spectra decreases. Okay. So for 7.2, this is the 7.2 microgram per liter, but fullerene for 36.0 microgram per liter, this actually decreases. On the other hand, of fullerenol, it can be seen that at lower concentration of fullerenol, that is 0.1 to 10, it actually decreases the fluorescence emission, but at high concentration like 0.1 to 1 decreases, but 10 microgram per level actually again increases. Okay. So there is a mixed property for fullerenol, but however for fullerene it shows a concentration dependent decrease in dityrosine formation. Now they have tried to see the dimer formation. So this is the monomer for the protein. Okay. Now oxidized RNase A, so this is a native RNase A. Okay. Now oxidized RNase A shows that a dimer is formed in this part. So the native RNase A has molecular weight of 13 kilodalton. And this oxidized RNase A shows another peak which is around like 25-26 kilodalton. Now in presence of fullerene, we can see that this band intensity decreases. However, for fullerenol, we can see that lower concentration, the band intensity is much lower. However, for higher concentration, it is much more. Now from this, it can be observed that although both fullerene and fullerenol can inhibit the dityrosine formation, but fullerenol is effective at lower concentration, but fullerene shows sort of concentration dependent decrease in dityrosine reduction. And now they have also used Malditoff experiment to show that the dityrosine formation, which is actually observed from Malditoff, that is like this is a native RNase A, and this dityrosine formation leads to dimer, protein dimer, which is around 27.6. However, the peak intensity of a protein dimer decreases in presence of fullerene as well as fullerenol. Now they have tried to understand the last thing is that how concentration dependent manner. So it as we have observed from the paper that with increase in concentration, the effectiveness of fullerene actually increases, but the effectiveness of fullerenol decreases, or rather we can say that fullerenol is much more effective at lower concentration. Now this paper is quite important from the point of view that it shows, okay, let's go back to the top on the paper. Now this paper is quite important since it shows that the fullerene and fullerenol, which exist in the form of nanoparticle actually, can interact with protein. So this is like a protein nanoparticle interaction, and in addition to that, it shows the fullerene shows different sort of binding behavior in the protein, and it can interact with the tyrosine residues. The next thing is that it prevents the dityrosine formation. So what we can say from this work is that in presence of fullerene, we can actually prevent the oxidative stress in the protein, but we have to be careful since fullerene is sort of hydrophobic and that is not pretty much soluble. So development of soluble product, or rather fullerenol, is quite important in this work. This paper has a short area where it actually shows how they have converted fullerene to fullerenol in a very short time, and like it should have come in some other paper also about the method, but this method is quite sort of impressive in the sense that previous method have used sort of phase transfer catalyst from organic media, but they have used like aqueous media to convert fullerene to fullerenol, and you can see it in the method material section. So this was it from the paper. So since this is the first video, so this might be a bit heavy, or rather I might be stammering a bit, but I will try to improve more in the next videos to come. Thank you for watching this one. Stay tuned and we'll discuss once again with some new and exciting paper. Till then, goodbye.